Understand the cause for plasmolysis. Understand the different phases during mitosis. Plasmolysis. Please finish the MCQs before studying our plasmolysis part. What is plasmolysis? Plasmolysis is the process by which a plant cell loses water when placed in a hypertonic solution, a solution having a higher amount of solutes than the cell. The actual process behind this is the movement of water outwards due to osmosis, resulting in the shrinkage of the entire cell. Plasmolysis is one of the results of osmosis and occurs very rarely in nature, but it happens in some extreme conditions. We can induce plasmolysis in the laboratory by immersing living cell in a strong salt solution or sugar solution to lose water from the cell. We use purple plant epidermal cell for experiment because they have colored cell sap which can be clearly visible. Osmosis is responsible for the occurrence of plasmolysis. Osmosis is a special type of diffusion that occurs when water flows into or out of a membrane, such as a cell's plasma membrane. It occurs based on the type of solution that a cell is in. A solution is a mixture that contains a fluid or solvent, usually water, and a solute that is dissolved in the solvent. When a cell is placed into a hypertonic solution, there is a higher concentration of solutes outside the cell, so water flows out of the cell to balance the concentration on both sides of the membrane. Since plasmolysis is the loss of water from a cell, it occurs when a cell is in a hypertonic solution. Conversely, when a cell is placed into a hypotonic solution, there is a lower solute concentration outside the cell than inside, and water rushes into the cell. In an isotonic solution, solute concentrations are the same on both sides, so there is no net gain or loss of water. Plant cells fare best in hypotonic solutions. This is because when plant cells are full of water, they push against each other to form the basic support structure for the plant and allow it to stand upright. Plant cells full of water are known as turgid cells. They exert turgor pressure on each other. The cell's rigid cell wall keeps them from bursting. Unlike plant cells, animal cells do not have a cell wall in addition to their cell membrane. When animal cells are placed in a hypotonic solution and too much water rushes in, they will lyse or burst. They fare best in isotonic solutions instead. Figure 1. Plant cells in different types of solutions. Let's see how water passes through cell membranes. The cell membrane is a semi-permeable membrane that separates the interior of all cells from the surrounding environment. The semi-permeable membrane allows some particles, ions, or water molecules across the membrane, but blocks others. Water molecules constantly move inside and outside the cell across cell membranes. This free flow of water has the very important consequence of enabling cells to absorb water. Plasmolysis and deplasmolysis. When a plant cell is immersed in concentrated salt solution, hypertonic solution, water from the cell sap moves out due to exosmosis. Exosmosis is the passage of water from higher water concentration to lower water concentration through a semi-permeable membrane. When a plant cell is placed in concentrated salt solution, water concentration inside the cell is greater than that which is outside the cell. Therefore, water moves through the cell membrane into the surrounding medium. Ultimately, the protoplasm separates from the cell wall and assumes spherical shape. It is called plasmolysis. When a plasmolyzed cell is placed in a hypotonic solution, i.e., the solution having solute concentration lower than the cell sap, the water moves into the cell because of the higher concentration of water outside the cell than in the cell. The cell then swells to become turgid. It is called deplasmolysis. If we place living cells in isotonic solution, i.e., both solutions have the same amount of solute concentration, there is no net flow of water towards the inside or outside. Here, the water moves in and out of the cell and is in equilibrium, so the cells are said to be flaccid. Making samples. Procedure. Take 2 glass slides and place them on the table. Take a real leaf from the petri dish. Fold the leaf and tear it along the lower side of the leaf. Cut off 2 to 3 centimeters of thin epithelial tissue and using a forceps, transfer them to a glass slide. Using a dropper, take some glycerin 2% solution from the beaker. Put 1 to 2 drops of solution on one slide. Place a cover slip over the sample using a needle. Observe them under the microscope. Change from isotonic solution to hypertonic solution. Put paper along a side to take out of glycerin 2%. Using another dropper, take glycerin 7.5% solution from the beaker. Put 1 to 2 drops of solution on the opposite side of cover slip. Place the slide under the compound microscope. Observe them under the microscope. Observation. After 3–5 minutes, we can observe that cells in glycerin 2% solution appear turgid, while cells in the glycerin 7.5% solution show plasmolysis. Conclusion. When plant cells are immersed in glycerin 7.5% solution or concentrated salt solution, water moves through the cell membrane into the surrounding medium because the water concentration inside the cell is greater than that which is outside the cell. Ultimately, the protoplasm causes shrinkage and assumes spherical shape. This is called plasmolysis. When a plant cell is immersed in glycerin 2% solution or dilute salt solution, the water moves into the cell because of the higher concentration of water outside the cell than inside the cell. The cell then swells and becomes turgid. Illustration for making samples. Transport of water